All right, hello, this is Antonio with Modern Cannabis and Modern Microbes, and I wanted to spend a little bit of time today talking about probiotic growing and its differences between other forms of growing. One of the things that I kind of hear most often when I bring up the topic of probiotic growing is really, what is it? If you haven't come across it before, you haven't heard the term, it can seem a little bit confusing, and I definitely understand that. So, to begin with today, kind of go off to a little bit of a slow start, I wanted to cover some of the basics and kind of explain why the term isn't necessarily as confusing as it seems. The term probiotic in the first place is defined as denoting a substance that stimulates the growth of microorganisms, especially those with beneficial properties such as those of the intestinal flora. When we buy probiotic food, we are generally buying them for their content of species like Lactobacillus, specifically stuff like Lactobacillus acidophilus, Lactobacillus plantarum. These are the same species that we are culturing when we make Lactobacillus cultures. So when we use the term probiotics inside the food industry, which is where you're most likely familiar with it from, and we use the same term in growing, it's because we're dealing with the same set of microbes. We're dealing with encouraging large sets of lactic acid bacteria and large sets of purple non-sulfur bacteria. Now, you may be wondering how we accomplish these things. Maybe, like I said, if you're new to the entire term, uh, you can do a lot of research if you are inclined to do so by using the term soil probiotics, because soil probiotics has been around as a term since around the late 90s, and it was originally defined that way. There's a lot of journals that still refer to it as SBO, which is soil-based organisms. And these organisms in question, strictly speaking, were those that would survive at first in the research, those that would survive from the soil all the way to our gut. When we would use like Lactobacillus plantarum, it's an acidophile. So if we culture carrots by having a high inoculation of this, or even just having growing methods that allow species that were already there to live and multiply very well, then you're going to end up with a high species, a high amount of it, high colony forming units on the carrots themselves. And when you eat those, because they're an acidophilic bacteria, they're going to survive all the way down to your intestines, and they're going to do what they do so positively for you. The whole reason you buy probiotic food, just by growing your food that way in the first place. And this is a pretty amazing thing. This is something that a lot of people, might find a little bit daunting at first, because they'll come across to ideas of like, well, how am I going to make sure, or how do I make sure that I have enough? It's really simple, and as you begin to do it, you'll see that the soil kind of takes over and it does all the work for you. So when you begin, just kind of follow along, do what everyone says, keep on a good schedule about it, and if you have questions, ask. There's a huge community of people that are more than willing to help, and one of the places you can look for that is online at Facebook, on my page, Modern Cannabis, or as my personal page, Antonio Bacchus. If you have any questions, I'm always happy to answer them. I know that it can be a little bit daunting at first, but overall, once you get it down, you'll find it to be by far the easiest way of growing. I mean, there's no more pHing, there's no more, worrying about the total dissolved solids, none of that. You just get into a schedule, you react to your plants, and they are happy. So let's talk a little bit more about what exactly is probiotic growing and what you might need to know about, like what's the difference between it and commonly, organic gardening. Well, if you're doing probiotic gardening, then the garden that you have is organic. But if you have an organic garden, it's not necessarily probiotic. When we talk about trying to culture large amounts of life, we're talking about biointensive growing. We're trying to make sure that every variable and input that we have to the soil is not one that detracts in any way from the life that's there in either fungi or bacterial species. So, one second while I have a drink of water. The things you generally will come across are, if you're an organic grower, you may already know about compost teas. Otherwise, you might be a bottled organic grower, and if you are, I really recommend heading this way. Bottled organics are not anywhere close to what you're doing with probiotic growing. There's a lot you can do that way, and a lot of people get stuff dialed in, and I won't say that I haven't had stuff that wasn't, really good for what they're doing, but it's all about input, and you're really never going to match up quite as well to what you can do with probiotic species and probiotic inputs, because with probiotic inputs you're looking at species and stuff like lactobacillus culture, like we talked about in a past video, that stimulates olivotolic acid and other fatty acid chains that are going to make sure you're ending up with higher end cannabinoid content, higher end terpene content, and that's a huge deal. On the backing up a little bit back into what you need to know as an introduction, if you haven't come across what compost teas are, then look that up. Actively aerated compost teas. You can find my recipes on my website under the Modern Methods page, or you can find information about aerated compost teas underneath the probiotic introduction section. The next thing you'll probably want to know about is sprouted seed teas. I know I've talked about that a little bit in the last episode, I believe, and that was to provide hormones, amino acids, enzymes, and other building blocks of the plant. You're looking at cytokinin, gibberellin, auxins, and all those. So endolysic acid, any of those different forms of natural growth hormones, and that's another really big deal. What a lot of people don't realize is that if you're using stuff like rooting powder or a rooting gel, you're using synthetic growth hormones, and there's really no reason to. I mean, you can use a small amount of aloe interfolate powder, which is very cheap. You can buy it over at buildasoil.com. Very highly recommend that website. You can use a little bit of aloe interfolate powder, a little bit of coconut water powder, which is another thing you can get there, and I use a little bit of my microbes because I have a very small amount of organic kelp ferment in it, and they have betaines, which will help the initial rooting process, and then you have the interfolate powder for giving it the endolytic-like compounds. In the coconut water, you have different novel forms of cytokinin, including zeatin, and all of these will make it so that your rooting process is very, very, very simple. So personally, I use rooterites. I mean, there's thousands of ways to clone. They all work well. Find what works for you. For me, I end up getting a little bit distracted with my business, so I like something that if I don't check on it that day, I don't have to worry like, oh, did the pump go out or anything like that. So I use rooterites and make a little bit of a soak. My exact soak is up again under the Modern Methods section of my website, but I make that. I put each rooterite in it, put it inside the tray, take your normal clones like you would, make sure you got your 45-degree angle, and then stick them inside there. Put the humidity tray over. If you're using a small amount of purple non-sulfur bacteria, you never have to really worry about algae building on the outside of the cubes. So I'll use a little bit of that. My source right now is quantum light, or quantum growth light. You can put the lid back on, put it under with a seedling heating mat underneath it. Some people like to kind of put those on a timer so they don't get too hot. I've really never had that issue. If it seems to be getting warmer than usual, just crack one of the two vents on the top. And usually, I don't know, 4 or 5 days for really vigorous strains and 7 to 12 for most strains. And I've got way more than enough roots to put them into their next container. You're good to go. It's very simple, it's very cheap, and it's nice because, if you don't check on it that day, generally speaking, you have no issues. I also like that purple non-sulfur bacteria in there for making sure that you don't get like powdery mildew. If you live in a high humidity zone, I live in a really dry place because I'm over in Colorado. I think my relative humidity generally stays around 30 to 40% unless I'm augmenting it in some way. So it's very easily leached to the air for me. I have lived in way more humid places, and when I do that, I do feel you need to use higher amounts of bacteria to make sure you're offsetting, the natural funguses that live there, the natural yeast that live there. I use a lot of my microbes and then a lot of purple non-sulfur bacteria to basically discourage those two things when they're young. When they get older, you can do, those, but in addition, a lot of other things. Out of products that are currently available, there is Big Time Exterminator, which, from my understanding of what the product is, which I don't want to say too much on, it's obviously organic. I use it myself, but it would definitely have antifungal actions. I mean, you're looking at a oligosaccharide, if I'm understanding correctly, what exactly it is. And again, I've not asked Austin Sherman what it is. I know that it was talked about on the show a little bit ago, the Adam Dunn show. Sorry for forgetting that. My memory is not the best today. I've slept way too long. But it's completely organic. Because of the complex that you're looking at, it would definitely have some antifungus properties. Mostly, from what I'm understanding, it's mostly through slight augmentation of jasmonic signaling. So you're going to have slightly higher jasmonic signaling when you're using that. We've kind of discussed a little bit on my website, and then I run a forum and a group about soil, and I've discussed a little bit with people there the salicylic response versus the jasmonic response, and I don't want to get too far into that in this video, but using stuff that stimulates jasmonic and salicylic response in unison, making sure that you keep them balanced, is very essential if you're trying to get very high trichome content and you're trying to get the absolute most that you can out of it, trying to get the most trichome to calyx ratio. So I do really like his product a lot. I use it on any of the clones that I get in from other people. I very highly recommend it. In terms of other forms of battling powdery mildew, like say you already have some, I've seen a lot of people be very successful with kombucha. Like if you're doing it yourself, I don't think I would go buy some from a store and spray my plants with it. I do not make I haven't made kombucha, so not here at least. So I don't do that. You can use fermented plant extracts for the most part. They all work pretty well. You're changing surface pH, and it'll kill most funguses. If you make it out of stuff that, excuse me, if you make it out of stuff that has a high potential for eradicating powdery mildew, like horsetail, giant knotweed. Giant knotweed's a really big one. There's a bioinsecticide / biofungicide product out in the market right now, and it's gaining a little bit of popularity with some growers. And I don't know if they know this or not, but it is just simply giant knotweed fermented plant extract. So if you have those near your house, go pick a few, make some fermented plant extract. You now have a very, very potent anti-fungal, anti-biocide agent. Actually anti-miticide. So it's pretty cool. Always wear your gloves if you're picking herbs where you're not sure. You don't want to confuse something. I've saw a few gardeners who were trying to make some fermented plant extracts with stuff on their farm, and they had hogweed, and you Google hogweed and you'll see what I'm talking about, but you don't want to touch that stuff. So wear gloves, be careful, people. Make sure you research things first. It's good to experiment, but experimentation should always be founded in what you know and what you would like to prove or disprove, because if you're just throwing stuff at the wall and trying to see what sticks, you're gonna not gain all the insights you could have. So that's not like a huge deal, but just something I wanted to share. I think a little bit earlier we were back a few topics. Sorry about that. I tend to be a little bit too genteel, but we were talking about sprouted seed teas, and to go on from that, other things that you're going to need to know about as a introduction into probiotics is stuff like lactobacillus serum, which is called a lot of different names anymore. You'll see it called EM1, which that's effective microorganisms, which was first defined in a paper by Teru Haiga, and that paper was the concept and theory of effective microorganisms, and it's freely available if you just Google it. It's very, very interesting. A lot of the results that he was finding, he could not continually find until he refined the process. So I've heard a lot of people be like, well, how come it wasn't what it was before? And how do we get it closer to that? And the reason that he moved away from that was because with stuff like Streptomyces and certain yeast populations, they would really outpopulate other things, and it caused his testing to become a little bit unpredictable. You can see if you Google his name and follow all the research papers he did, why he slowly went towards the just lactobacillus and purple non-sulfur bacteria. Those are really the powerhouses of what's going on. Now, that doesn't mean that there aren't other species that are hugely useful. There are, without a doubt. It just means, don't necessarily try to recreate what was done originally when we found what was most important in that. Do the research and find out why you want each species in there, and find out if it's going to cohabitate with other species that you are relying upon. There are a lot of really good forms of Streptomyces. I would like to say that real quick. But on to EM1. When we make lactobacillus culture, we a lot of times just call it EM1. You'll see it AEM1 because it's activated by the end. Once you've added the molasses, you've added a sugar source so that they can sustain themselves and continue to multiply to a certain point. And that's why you can only use them for so long. If they exhaust all the molasses, then they'll begin to feed on themselves, and the amount of acids in the solution will become too intense and it won't be useful. So you can either continually add very, very small amounts of molasses at every few months of having it, or just make more, which it's not really that expensive to make. If you're not familiar with it, you're just buying the best quality milk you can get with the very highest amount of milk fat. So, cream would be technically better for this, but you can use milk. I buy just cream on top milk because I don't have a dairy farm near me that's they won't. Well, I do actually have a dairy farm near me, and I asked them if they would sell me raw milk or anything like that, and they will not do it. So because of that, the best I've been able to do, the highest fat count, was going down to a health food store, and I found some really good stuff where the fat's still on top and you have to shake it to get it all homogenized. And the lactobacillus cultures from that were incredibly potent, and you could see the difference in colony forming units underneath the microscope because of the higher fat content. There may be a curve response to that where, if you're going so long, it's going up, and at a certain point it's going to level out, or perhaps even go down. I don't know. But so far, that's what I've been able to find in terms of usefulness. You're also going to need to know about fermented plant extracts. I've talked about that a little bit in the last video, but I didn't cover one question that someone had, and I wanted to cover that real quick. They asked, if you used a root powder inside of a fermented plant extract, is that okay? When they said that, I immediately thought of maca root, and I have done that, and it works very well. Don't make a fermented plant extract solely out of something that's powdered. Add it in addition to the very fresh fruits and veggies that you're using. But yes, it does work. It definitely does. And I recommend experimentation with maca root in particular. It's harvested at a height where they don't need to use pesticides, and it contains an absurd amount of the minerals we're using, amino acid complexes, all of that. It's got some very unique properties, and I highly, highly recommend people kind of play around with it. I've not ever had any negative effects from any of the experiments I've done with it so far. So if you do know anything or have tried it yourself, just let me know. I'd be very interested. Another thing that we talk about a lot, we mention systemic acquired response or systemic acquired resistance. What this is, it can be thought of as kind of like the plant's immune system, or it's more correctly, it's known response to specific stimuli so that it can keep invaders in terms of bacteria, fungi, pests away the best that it can. So they've done tests even as much as you infect one plant with bugs, and it's going to release signals and hormones via its roots that are going to communicate through a mycelial network if they're outdoors, and that plant near it is now going to begin to produce hormones as if it was being attacked by these bugs, so that it can begin building up defenses. And this isn't the only way of doing this. This is a huge thing. When we talk about jasmonic response and salicylic response, we're talking about the two more well-researched pathways when it comes to acquired resistance. And we have induced acquired resistance, and shoot, I'm spacing on the other one, but it'll come to me in Just a second. Oh, there it is. Okay, cool. So we've got systemic acquired resistance and induced systemic resistance, and both of those can be biotic or abiotic. So we talk about using stuff like aloe interphyllate powder to use higher salicylic compounds to increase our SAR. So when we talk about that, what we're saying is we are increasing the plant's defense networks for specific pests and specific stimuli. Recently, we have found out in the last few years through studies on Arabidopsis, which Arabidopsis is not a glandular trichome, if I remember correctly, unlike what we are, but, or rather what we're studying, but it is incredibly similar in behavior in terms of creating sugars, acyl sugars, and stuff like that. So a lot of the research has been able to come over really well. So they were doing studies on that, and they were doing studies on the frost plant, the ice plant, and it was near jasmine, and they harvested a bunch, and they found out that the methyl jasmine going over on the wind was able to increase trichome of those plants, if I remember correctly, at right around like 65%, and then it reduced the overall plant size by about 30 to 35%. And this was something that they had never encountered before. So all of a sudden, all this studying began to happen on methyl jasmonate, and methyl jasmonate was this main plant exudate that was coming from, chopping jasmine, or, it's even contained very high in jasmine's essential oil. And with cannabis, there was quite a bit of experimentation done, and people have found, yes, it totally does increase your trichome, but it also reduces your bud mass. So there's something very important about keeping these two forces balanced, because the more you express the jasmonic pathway, the more activation energy it requires for the salicylic pathway to be activated, and vice versa. So using these factors that we are to stimulate either the salicylic pathway or the jasmonic pathway in tandem is really important, and it's something I want to talk about deeper in a more advanced video. I know I'm a little bit getting into some of the more deep stuff in an introductory video, but it's a little bit too much fun. So I always want to talk about that. So back to what we were talking about again, that's just the SAR response. There's a lot of papers out on it. You can look up systemic acquired resistance, you can look up systemic acquired defense and induced systemic resistance, and you're going to find some amazingly useful stuff. You'll start to understand a little bit more about the plant and why we avoid pesticides, why we avoid fungicides as well to a big degree, which is something I want to talk about in another video. All of us are trying to, have healthy mycelial networks. We're trying to have healthy fungi networks, and then I see stuff like activinate be used to eradicate PM, which is a biofungicide. And of course, if you're going to be using a biofungicidal agent, it's better than using something that's a straight pesticide, but that one is particularly intense and wide spectrum. So the more we're able to kind of narrow these things in, the more we're able to find specific treatments for powdery mildew. And really, if you're getting into IPM, which is integrated pest or integrated pathogen management, you're not going to be dealing with that as much. Once you get on protection, you won't have to worry so much about treatment. And that's a huge thing. That's that whole ounce of prevention is worth a pound of cure. So actually, speaking of integrated pest/pathogen management, that brings us to the next thing you need to know in Introduction to Probiotics. This is how we stay away from having predators, pests, all those things in the garden. It's not a matter of, hey, what do I do now that I have it? That means you have, waited for, that to happen to you, and you don't want to be that way with your garden. If you are that, if you feel like that right now, and we've all been guilty of it, don't get me wrong, like every single one of us, so don't feel bad about it, just get in the habit. It's as simple as making a few, timers on your phone or whatever you have to do, and making sure that you are there to get your plants sprayed down. When I say sprayed down, the stuff we use are essential oils. There's a essential oil blend over at Build a Soil that works incredibly well. Neem and Karanja oil. I like to avoid using these for the most part. If I am getting fresh clones in, I will give them Big Time Exterminator, Neem and Karanja oil, essential oil, microbes, and I'm probably forgetting one or two things that are usually in my mind. Oh, Natural Mystic. I like Natural Mystic from Dragonfly Earth Medicine because they use a lot of those really, really good probiotic plants that have natural flavonoids, bioflavonoids, phenolic compounds that help discourage predators and pests from continually coming. So I'll use those. I will use those a lot of times with an anaerobic ferment, or just an anaerobic tea of insect frass. The On Frass website will tell you how long to do it, and it's like 3 to 4 hours. So it's a great way to kind of do something that doesn't require a lot of like work or planning ahead. I mean, you do the Natural Mystic, if I remember correctly, is another anaerobic one. It goes overnight, and then 3 hours before I would finish it, I add my insect frass. Once that's completely done, I can add the rest of everything else because nothing else will be anaerobic. So I'll open that up. I'll add my, at this point, generally my essential oil, my microbes. Make sure that your water is somewhat warm if you're adding neem and karanja oil. That's really important. It's actually important with the essential oils as well. Try to keep your water warm in general. All of you, if you haven't been doing this, try to keep your water right around like 83, 84 degrees constantly if possible, and you will notice that your roots are going to be a lot finer and a lot healthier, and the more fine that your roots are, the more they're going to spread out and the more that they're capable of picking up very quickly, and the higher transpiration rates that they have, and higher osmotic potential. So you end up pulling a lot more, and that's part of why we use these really good bacteria. It's part of why we use quality water, good well water, using a reverse osmosis machine, any of those things. Make sure your water is good quality. You don't want chloramine sitting in there. Chlorine or chloramine. Some people use tap water and bubble it out over 24 hours, and I know that that does get out chlorine, so that's not an issue. I still prefer reverse osmosis myself. I like to make sure that I'm not getting like slight amount of magnesium or any of those from outside sources, so that I know exactly what I'm controlling. But I came from a background of I like to keep everything very listed. I make a lot of documents and journals and all that. I apologize. I have to take so many drinks of water. I'm like constantly cotton mouth. But here's a Grow Kashi. So one of the things that you're going to need to that you will eventually come across are a bunch of the probiotic products that are endorsed through the PFA and through the rest of us. Myself, as Modern Cannabist, I've constantly endorsed Grow Kashi and other forms of Kashi as well. I use Grow Kashi and O-Kashi in my garden. I love them both. They work wonderfully well, and they have slightly different contents. The Grow Kashi is a little bit has plum extract, molasses, and azomite, and then O-Kashi has high fermented plant extracts. So I do really like both those products. Getting familiar with Kashi is a little bit of a weird thing. When you get it at first, it's this like grains. It's like you can kind of hear it in here. These grains have been inoculated with lactobacillus. So when we do the anaerobic ferment like that, we're making a lot of lactobacillus and some streptomyces, and these are really wonderful species to be able to culture so easily and so quick. This overnight anaerobic fermentation. The other thing it can do that's really cool is advanced decomposition and advanced composting. We can make a topsoil layer and water it down, water it with a compost tea, water it with fermented plant extracts, any of those, and you're going to see it really take off very quickly. I like to put a top dress down where you have higher earthworm compost content or higher compost content, and then put them on top of that. You water them down, and then you cover it. And this is really important. Please cover it, otherwise it's going to dry out, and it's just going to make it very, very, very difficult for you to water, because you have this like really thick layer that gets really hard and doesn't want to let water through. And if you do that, wait for it to kind of dry out, break it up, and you'll be able to pretty much just remove it and take it off. I've done that, so don't feel bad. We've all done it when we first try this. So really cover it. It's not that big a deal. I've tried all sorts of different things for covers. You can use plastic contractor bag. It works really well. Just get yourself an X-Acto knife, cut out some covers where they've just a circle and a slit so that you can put it up to the stem, and that will keep the humidity there. Once that happens and that pseudo-hyphae layer starts to take off, and specifically you're gonna see a little white fuzz. Don't freak out. That's pseudo-hyphae. A lot of people will be like, hey, that's mycelium, right? Not quite. It is pseudohyphae that's interacting with the mycelium. It's tying itself into that network, and you end up with straws from the top of your soil for, you know, watering, composting, any of that stuff that will directly deliver themselves into that rhizosphere now. And that is amazing. It makes you use less water because it's not transpirating out as quickly. The pseudohyphae layer is going to absorb the water and try to hold it in. So you can reduce your water usage by about 1/3, which is a huge deal. It will make your plant healthier. You have a healthier rhizosphere. You have like more direct control about how to get stuff into it. So I hugely recommend it. It's made a huge difference in my garden, and I do think it'll make a big difference in yours as well. If you're doing anything else, I don't really think that there's a lot else to cover with probiotics in terms of differences between organic growing, and just say, you know, kind of stay away from a large amount of products just because it's just not necessary. Look at stuff that can be amendments. If it can amend your soil, it can be a top dress, it can be a tea. That's an important concept. Also, if something works as an IPM, like I use neem and Karanja meal. This is something I think a few people have kind of talked about, and I've tried to talk about a little bit, but neem and Karanja meal are natural IPM, but they're also slowly breaking down sources of nitrogen, phosphorus, potassium, microminerals, especially in the Karanja. These are essential. I use them as my main amendments. I use them in top dressing, and as my main amendments, it means that the stuff that is the core of my nitrogen, my phosphorus, my potassium is also acting as keeping bugs out of my soil. And I use every 3 weeks I inoculate with nematodes. This is important. Nematodes are part of what's going to help keep you from ever getting the fungus gnats that seems to be a constant problem. As long as you have, like I like to do the Arabico organics and then the triple pack where you get all 3 species. I do that because, you know, you never really know what you're going to encounter. But these beneficial nematodes will puncture eggs and larva sacs and stuff like that, and they secrete an amino acid that's like protease and, if I remember right, protease and I want to say bromylase, but it will literally liquefy the contents of that, and then they just slurp it up, and you end up with amino acids in your soil that your bacteria love and that your plant loves, and you don't have, you know, your soil is not just defenseless. If a bug does manage to get in, it's not going to, you know, instantly turn it into its home. That's how you help keep yourself from having a problem later. If you have, you know, nematodes in your soil and you have this thick mycelial, or excuse me, I just made the mistake that I try to correct so often, but if you have that thick pseudohyphae layer on the top, then you're not going to have fungus gnats constantly getting in and able to lay a bunch of eggs. And that's a huge deal. Another cool thing is every time you use anaerobic insect frass, you're having a high amount of chitinase enzyme, and that's going to dissolve a lot of egg sacs as well. And if you're doing that, what I generally recommend is doing that and then the next day inoculating your nematodes, because it will also dissolve the outer lining of your beneficial nematodes, eggs, egg and larva. I forget exactly which form they reproduce, if it is egg or larva, but either way, I do know that when I was reading that it does break down their population slightly as well. So use that, and then the next day inoculate. If you stay on top of that, and that's not really a very expensive thing, you'll also notice that because they help with other bacterial species that are in there, you're going to end up with healthier plants. You're going to have a really thriving micro community that can support itself a little bit better. I also use Ladybugs. Not everyone has to do that. That's up to you. The only reason I personally like it is because they end up lasting a long time. I don't know if that's because I just have, I guess they last like 4 or 5 days, which I suppose really isn't that long, but for an insect's life, that's pretty decent. Most insects don't live all that much longer than a week, so it's not bad. And then their subspecies will usually flitter around for about, you know, another 5 days. Most of the insect carcasses I can throw in my soil and they'll break down into natural insect frass. So I kind of do it for that. So I don't know if that's kind of a weird thing to do, but they end up with protein, amino acids, you know, chitinase enzyme just degrading naturally into your soil. And it's a great way to just have something natural that if there are ever bugs or anything, they're going to eat them very quickly. So I'll usually release them next to all like new clones I'm bringing in and stuff like that, and then see how long they take to kind of diffuse through the rest of the room. Usually within a day, they're mostly up inside my lights, and then they die in, over the next week, and sweep them up and just throw them in my pots. And my kashi layer on the top loves to break them down very quickly. All my plants always end up taking off real nicely afterwards. So it's something that I do. You guys can look at it if you want to. If you don't, there's no worries there. Like I say, there's no one perfect method. Everyone can just share what they know, and what works for them, that's good. The reason I suggest more people go towards probiotic growing is not necessarily because, I mean, I do think it is the best method of growing, I will say that, but the reason I do it is because I'd like to see less runoff. I'd like to see less damage to our soil and less damage to our planet. This is actually a bigger problem than what I thought it was. I've been reading a lot of books that talk about the rate at which we have naturally composted matter, natural organic matter, and the rate at which we use soil for farming and major agriculture. But 2 days ago, there was a study that was, or at least I became aware of it 2 days ago. I think it might have been 4 days before that was released. But they did more up-to-date calculations of the amount of organic matter we produce as human beings across the entire planet, and how much we're using as an agricultural industry, and we are not sustainable within 70 years. So this means that if we continue at the exponential rate at which we're growing and the exponential rate at which we're consuming, in about 70 years, we're going to end up with mostly synthetic/hydro methods, because they're not going to have all this really good organic matter and organic soil that we're going to be necessary without destroying habitats, various places that we're already kind of endangering. So I'd like to not see that sliding scale continue. I'm hoping that we can make it from exponential to at least decreased, and then over time corrected. I advocate this because, especially with us as cannabis growers, the newest way we're demonized is, hey, look at this. They're going to have huge amounts of phosphorus inside the runoff, inside our watersheds. We're going to have huge amounts of nitrite that are coming from it. And they could be right if we let them. I mean, if we continue with hydro methods where that's what's inside of our runoff, and, we're just freaking washing that down the drain, then yeah, that is a serious issue. If we do stuff like using purple non-sulfur bacteria, using lactobacillus, there's actually a bunch of species. A big reason I put bacterial solubilizing bacteria inside modern microbes is because the ways that they degrade these compounds into being plant available again means that if you have like overworked farmland because you've been doing way too much salts, so your phosphorus levels are huge and they're still inside these groups that are freaking, the plants can't use them, nothing can use them, so we consider that oversalted soil. They can be broken down by these bacteria. They can be broken down back into states that the plants can use, absorbed back out, and we can go back into using a natural amount, naturally from the compost, looking at stuff like fish compost, stuff like that, and not have this issue where we're building up to salt levels that we can't support and then waiting for bacteria to leach it down enough that it's usable land again. I mean, we've kind of taken the most, in a way, barbaric way to do growing at this point. We're willing to pillage the land, and when I say that, I mean that because when we're doing like nitrite, pulling it from places, we pull it from mountains and stuff like that. We do blasting caps and large nitrogen factories out in very dry environments so that they don't end up exploding, and we've all seen what happened when some of them do, and that footage is on the internet, and if you haven't seen it before, it's crazy. There's no reason for this. Like, we don't need these. They're used in explosives and fertilizers. If we're talking about fertilizer usage, we simply don't need it. We have bacterial nitrogen fixers, and then of course it's going to take multiple forms, and then we have stuff like alfalfa, we have stuff like soy, any of these things, even hemp. I mean, the plant that we're growing itself, it contains a decent amount of nitrogen in the end. That's a very green plant. We grow it in methodologies that try to reduce the amount of overall content, which is something that I'm very interested in seeing how it progresses as we continue as growers and our testing increases. But there's not a reason for us to continue the way we have in terms of agricultural production of these products when there are better options available that aren't going to be using up this, that allow us to recycle and to do less work. I mean, why continually go and do tilling over and over and over, destroying that fungi network, when instead you could rip a plant out, put the next one in, top dress, and keep going. I mean, yeah, you're going to have to cycle out what happens in that pot with specific crops. With cannabis, I haven't had that issue. At that point, it's more about making sure that your stuff's never building up in terms of like calcium to phosphorus level, that doesn't ever become unbalanced, or excuse me, calcium to magnesium, and your phosphorus level, making sure that never becomes too high or unbalanced. It's amazing the actually pretty relatively low amount of phosphorus that cannabis needs to be very full, as long as you're supporting heavy hormone and like hormone content. If you're looking at stuff like auxin and all those, and you're continually helping it by giving it, all these enzymes are lessening the activation energy of the plant's potential mechanisms, right? So you have your barley sprouted CTs, and all of these have huge amounts of amino acids. That specific one, when you're using barley, generally speaking, you're using it for amino acids. Amino acids and enzymes are going to lessen the activation energy required for you to do metabolic processes or produce or divide cells or any of those things. So you're giving them those, and then at the same time, you give it something like corn SST, right? So your corn SST, you're getting cytokinin, you're getting small amounts of zeatin. Put that in there with a little bit of your coconut water powder and your aloe for a salicylic response, and all of a sudden you're literally reducing the activation energy required. You're giving it the necessary hormones for it to do the growing, for it to do the elongating, for it to focus on its fruit, and you're lowering the energy that's required for it to do that. So you're going to be getting as much as you can possible in terms of just purely biological systems, because you've honed in those variables. You've taken down from, oh, well, they have to produce this much of this specific amino acid complex for it to activate and for it to do that next thing. Well, we've supplied most of that, so now it's a very, very small amount, and by cutting down each one of these things that the plant has to do on its own, it's constantly just focused on what we need. And we can do this further and further the more that we're understanding about it, and the more that we learn how different plants want different things. And that's another really important thing. One of the reasons that we see the commercialized industry kind of be so different than what we can get from home growers is a lot of times the care that they can take from plant to plant. Now, if you've ever grown like a really old heirloom species, or even just like, it doesn't even have to be that old, Apollo 11, that's a really easy option. Apollo 11 is incredibly lanky, and it can be very, very fussy when it comes to nutrients. It requires very, very, very little. I found that when I've grown even hybrids with it, the stuff that's Apollo 11 dominant, it would get like floppy. It would get kind of crazy if you gave it a tea. It just really didn't seem to like it. It would slow down a little bit and just be kind of unhappy. And when you gave it barley SST and corn SST as only the only things you ever gave it, so just let it do its own thing, it's beautiful, like big, beautiful buds, super easy to grow outside of the staking. The staking has still always been difficult with that plant, but it's worth it in the end. And you can start honing these plants in terms of how they express themselves and how much you're getting from them. I know a lot of people get concerned when they're coming from hydro. Well, how am I going to equal these same amounts that I did before? It's totally possible. It's actually not entirely that hard. It's just a matter of looking at the plant in a new way. You're not looking at it as, oh, I'm just going to be giving it nitrogen, I'm just going to give it phosphorus, I'm just going to give it, all these primary and secondary nutrients. We're looking at the entire scale. We're looking at whether or not you're giving it the amino acids it needs, the minerals it needs, how these interact, how the bacteria and fungi interact. And that shouldn't seem as daunting as it might be. It's actually really easy. And as you follow what you're seeing other people do, you will see your soil take over and show you and teach you a lot of what you need to know. You'll put something in and your soil will respond accordingly and your plant accordingly. This is really what it means to be doing probiotic growing. It means trying to always maximize the amount of life so that it can lessen the amount of work that your plant has to do, which is what's happening in nature. If you wonder why the forest doesn't have to be pH to be as beautiful as it is, it's because these species, when working together, can already control this. They know what they want. They know when there's a specific amount of lignin and carbohydrates inside the soil that the fungi can, continue to take off. The mycelial network's going to expand. It's going to break those things down. That xylanase enzyme that's going to be produced by those networks are going to break that down into a bunch of simple sugars that the bacterial species are going to feed off of and going to continue to get larger. if you want to learn more about these things, I really highly recommend, if you like to read, Teaming with Nutrients and Teaming with Microbes. Teaming with Microbes comes first, then Teaming with Nutrients. You can see I have a lot of notes throughout them. Get copies of them. It's not a small deal. It's a great book. They are very well written. They're very simple. They're written as an introduction for people who are coming from basic gardening over to this style. And there are a few things in there that we might not necessarily always agree with. But they are 100% wonderful books. They're very well researched. They're written by incredibly intelligent authors, Jeff Lowenfils and Wayne Lewis. And if you don't have them, you're doing yourself a disservice. To get further back onto the topic, if you are unfamiliar with organic growing, you might not know what top dressing is. That question just came to me from a user here. So I wanted to cover that real quick. Top dressing is just making a mix and putting it over the top of your soil and watering it in. Here's kind of the way I see it. When you have a well-amended soil, you can do composting, which is like you want the action overnight. You can do top dressing, which is you want the action over 7 to 12 days. And those are really your scales. So like right now, I'm doing a kind of a big garden, and I want a lot of time to trim the plants, to top them a lot of times, train them out, low stress train them, get up 2 levels of terracing, and make sure that they're very well supported before I go to flower. So I'm just going really slow with them. I haven't done a compost tea yet. I haven't needed to. I've done a few anaerobic teas and mostly just top dressing, because if they look like, they need a tiny bit of nitrogen, then I'll just throw a little bit of alfalfa meal on the top along with some insect frass. Generally speaking, I like to take earthworm castings. If I have compost on hand that is extra, I'll do about 80% earthworm to 20% compost, and then I'll put whatever it is on top dressing. And anytime I add something for top dressing, I like to add a very small amount of basalt and a small amount of gypsum. And that's I guess I shouldn't say every time, about every other time. You can see my exact list on my website, but it's pretty near every time. I do this because basalt is a really quick way to start bringing calcium into your soil, and the gypsum is a nice medium length way, and the sulfur that it has with it will bring out the odoriferousness of your plants. If you guys haven't tried I know a lot of probiotic people really disagree with me on this, and that's all right. Like I say, there's no one right way. I definitely don't think that anyone really has it figured out with this plant yet. I think we're all just reaching towards the same thing, and that's just doing it how nature already did, and seeing the best that we can get the expressions out, and how to replicate, specific biological actions that we were getting in the past with chemical fertilizers at a higher rate inside the plant with organic means, just by understanding the plant and its biomechanisms. So I know a lot of people don't feel like you should flush. They'll say, if your soil is really well amended and you're doing the whole no-till thing, then what are you really doing by flushing it? And that's a really interesting question, and something I've really done a lot of thinking about, and I don't really know that I have an answer that I want to go with completely yet. But for the most part, if you are putting a large amount of water through it, and you're not top dressing, and you're not adding in additional things to the water, you are continually having what's exactly near the roots that and the hyphae is getting washed away a small amount over time. At first, no, it's not, because of course you've done a good amount of top dressing, and perhaps really I'm just doing something for no real reason. I mean, I guess I should divide a room and half of it not do my tapering at week 6 of no longer providing any top dresser other than gypsum and some rock dust, some calciums, and kind of find out exactly what the difference is between the two. But I like to do dissolve gypsum into my gypsum and oyster shell flour into my week 6 water, and then just gypsum at 7, 8, and 9. And the reason I do this is that continual sulfur is known to stimulate basically the fatty acid biosynthesis pathways that are associated with a lot of terpene production. Not all terpene production, but a lot of it. And so with many, many species, you will find that just doing that continually will end up with more odoriferous plants. If I were to kind of approach something that I've read a lot about, if you've ever used the product Terpinator, tons of people have, you're buying citric acid, azomite, and gypsum. That's all it is. And I kind of feel like the azomite's there just to throw people off with the high amount of that toxic compound. I can't remember exactly what it is. It's why a lot of people don't use azomite or arsenic. There we go. So I think the whole high amount of arsenic is really just the azomite. But what you're buying is citric acid in a water solution. So you have an acidic solution and you're adding gypsum to it, which is going to make it so it's more easily available and you're going to have a slight amount of natural chelation. So if you've been using that, hey, whatever, no biggie, I don't judge people for that, but just know that you can do it a lot more simply just by adding a small amount of your fermented plant extract, because now you have an acidic solution. In this case, you're looking at like acidic acid versus citric acid, and adding that to your gypsum, letting that sit for a little bit, you know, say like 4 or 6 hours, and then adding that to your water and then using it. And you're going to have a more highly available amount of gypsum, which is going to give your plant that same sulfur that they were going to be getting either way, and more easy, small amount of calcium. If you try this and you do side by side, you will see your plants are a lot more odoriferous in the end by utilizing a little bit of gypsum. So try it if you like. I encourage it. If you feel like you've found something that works better, I'd be very interested to know. Like I said, there's no one right way. So it's just what I do. So I hope it's helpful for everyone else. And I hope you enjoyed this video. I'm going to open up the question thing again and see if there's any more so far. I don't think so. In fact, actually, I'm kind of not seeing where. Oh, wait, there it is. Question and answers. Boom. No. Oh, there we go. What is the bacteria from EM1 and like products derived from? Good question. When we talk about lactic acid bacteria, generally speaking, we're using something like a carbohydrate solution. So you use like rice. You can really use any carbohydrate. That's all you're really doing. You're making a carbohydrate solution, and then you're allowing it to spend enough time to get high colony forming units of many different species. And then you add milk. And the reason you do this is because all the lactic acid bacteria are going to take over, and they will continually put out they're going to change the pH of the solution. And as it begins to go down and become more acidic, we're going to see all these milk fats begin to coagulate and form together. And this will form that big ass like whey cheese thing that's in there. It's actually really probiotic and good for you. I'm lactose intolerant, so I can't eat it, but I give it to my dog. She loves it. It makes her super happy. It makes her all energy, you know, have a very high amount of energy. But as that begins to happen, these species continue to take off because they are supported by this environment. It's what they want. And it detracts from the environment of all the other ones. So over a short amount of time, you end up with mostly just lactic acid bacteria in it. And then you add the molasses, and this causes those to be able to be sustained. They eat off that, and they stay at around that level. They'll increase and then decrease in small amounts until they run out of sugar, and then you generally drop in terms of pH hugely, and then they all start to die, and it smells all funky. With the original recipe of EM1, they were also including Streptomyces, which is a form of yeast, purple non-sulfur bacteria. Purple non-sulfur bacteria, the predominant forms that we're getting were Rhodopseudomonas palustris, and there's another palustris that I'm not thinking of at the moment. But both of these purple non-sulfur bacteria are cultured from pond water. And I know that on Wikipedia, it says that they're found in high amounts in earthworm castings. So I want to talk about that real quick. I went and reviewed a bunch, like there's no reference for that. It's not cited. And if you go read any of the things cited on that page, the place that they're pulling that from is MicroWiki, unless MicroWiki pulled it from them. Either way, neither one of them sources it. So I went and just searched earthworm castings PNSB, and all the results were showing that pretty much there wasn't an overwhelming amount other than there was from background amounts. So I don't really know where first someone wrote that there are huge amounts in earthworm castings, but it's actually a pretty like minimal amount compared to like even your compost has slightly higher than what the earthworm castings did. If you want a higher amount, so you're trying to go for a higher amount of bioremediation, you're trying to utilize that for anti-algae, whatever you're using it for. They also use it in the fish industry hugely. Koi ponds, I know if you're buying bacterially based koi homogenized, whatever they call it. I don't do aquariums very much. I just came across it in my research. They use it for keeping algae off in those as well. So it's used in aquaculture, it's used in aquariums and all those. It's cultured from pond water. There's a bunch of really good websites about that. If you've gone through Biology 103, I think it was where it is, where they start teaching you how to use most of like the incubation machines and all that, then you will be pretty familiar with most of these. Like the one that I really liked was hosted. I can actually check that out real quick. I keep a ton of bookmarks for all these different types of things, but give me just 1 second. So that would be under botany, microbes. There we go. So it's mbio.ncsu.edu. And sorry for the weird link, but you can usually find it just by searching Microbial Diversity Lab, purple non-sulfur bacteria, and they do go through enrichment and isolation of purple non-sulfur bacteria. Another really cool thing about purple non-sulfur bacteria, if you're looking up peer-reviewed journals and you're wanting to just kind of find out more about how it works and get a better understanding of what's happening in your soil, it is actually being used these days for bacterial bioreactors. And if you've ever taken the enzyme CoA, coactive enzyme 10, this enzyme can be produced in huge amounts by purple non-sulfur bacteria. So there was this really cool paper I read where they were doing fermentation-based culturing of purple non-sulfur bacteria and then tweaking it to try to get the highest amount of the coenzyme Q10 that they possibly could, so that they could harvest it, obviously, for making soft gel pills or whatever. And it's just really fascinating. It shows you a lot about how it's cultured and how it could be done actually on our side for a smaller scale, very, very easily. Most of these places that are doing them these days are Malaysia, Asia, places like that, because like I said, they are hugely used for koi and all that. So they were known about for a long time because of that. So if you go to some of these like aquaculture websites and stuff like that, you'll inevitably come across something that'll say like Pseudomonas cleanser and stuff like that. You're buying PNSB at that point. I know that there's been some just straight untrue things written about PNSB lately. I saw someone, I won't say who, but I saw someone saying that they had developed a patented process for preserving Rhodopseudomonas palustris. And I want to talk about that real quick. This is nonsense, like hardcore nonsense. If you want to find out the first people who did it, you can look at Hawaii, Japan, Malaysia. These people have completely written up papers, detailed research notes on preservation, on inoculation, on shelf life. All of those patented process were stuff that were taken from those papers and patented here, or there is no patent, which I haven't been able to find one, so I'm more likely leaning towards that. These aren't that difficult. The difficult part is actually isolation and making sure that you have large enough grounds basically to keep them happy, keep them propagating, make sure you have all the stuff on hand to detect how many colony forming units they're at, what the amount of amino acids and oligosaccharides, polysaccharides, etc. are within the solution to see how they are going to have specific PNSB colonies either wane or wax, So don't be discouraged when you read stuff like that. It's not, in the end, I think most people just don't really don't want most people to know that. I don't think that they want the competition that could come from passionate people that would prefer to do a little bit of learning and set up a lab somewhere. So don't let that discourage you. I think that kind of covers all that's inside of EM1 that people really need to know about right now. Is there any other questions before I finish up this video? And then here in probably 20 to 30 minutes. I don't know, somewhere in that range. I'll probably do a second one and go over some more advanced topics, but I wanted to make this one first because a lot of people asked me to cover what are the basics of probiotics, what makes it different than organic, how do they come into this, and if anything I've said today has been unclear, or if you just want more information, you can check out my website at modernmicrobes.com, look under the probiotic introduction section, or you can look at the Facebook page for Modern Cannabist. You can contact me, Antonio Bacchus, and I'm super, super, super happy to answer questions. If I say something wrong, which actually on that note, real quick, tricannatol is the hormone in alfalfa seed, tricannatol. I said that wrong in the first video, and I was corrected, which I really appreciate because I do very often. I don't have someone to talk about it with, so I just read all day, and I might very well mispronounce a word. If I do, just let me know. I'm happy to hear it. I'd prefer to know. So if you watch that first video, again, that's tricannatol. So if you find any information to be incorrect, or you just want to ask me some questions, just shoot me a line at any time. I'm happy to answer. And I hope you all have a wonderful day.